Okay, folks, let's get started. Happy Tuesday. Tuesday, your favorite day of the week? No? Is that coffee or is that something else? You brought the coffee? Yeah, well. Yeah, well, it kind of gets it in the system a little faster, right? So, I wish I had something witty to say to get everything started, but I don't, so you'll have to just put up with my usual stuff, I guess. What did I just do? I moved something. Top right, next to the microphone. There we go. Okay. We're moving rapidly through lipids and membranes. I've only got one more thing to say about them, and then I'm going to say a few things about vitamins, which, of course, are lipids, at least the fat-soluble vitamins are. And a brief word about prostaglandins, and then we're going to talk about nucleic acids. So that's the plan for the day. I hope I've been able to plant in your heads this notion that membranes are good barriers, and that to work around that barrier, cells have to have some proteins that help them to function properly. And you've seen in here a couple of different membrane proteins that are involved in moving things into. There are also things that move molecules out of cells. And in fact, on any given cell, there are thousands and thousands of these proteins that I'm talking about. So it's not rarities that we're talking about. There's a lot of different proteins that are involved in doing some of the things that we've seen here. So that's important to keep in mind. In fact, some organelles, if we look at mitochondria, which are, of course, organelles within our cells, mitochondria and other organelles have their own membranes because they're membrane-enclosed structures. The mitochondrial inner membrane is one of the most protein-rich membranes in the world. It has about 90% of its weight protein. So it says that, in some cases, the protein component of a membrane can be quite impressive. Okay. Well, what I want to say next is one other consideration with respect to membranes, and that's something that you've all heard a little bit of something about before, and that's LDLs. So if you've ever had any issues with cholesterol, or you know someone who's had issues with cholesterol, you've heard of the term LDL. We'll talk about LDLs later in the term, but I want to point them out right now for one important reason. And the important reason is that LDLs have a different way of crossing the membrane than other things do. All right? So LDLs are complexes. They look little things that look like this, at least schematically. And what they carry within them are water-insoluble things. They carry fats. They may carry fatty acids. They may carry fat-soluble vitamins, and they certainly carry cholesterol. That's the context in which most people hear something about LDLs. Okay? Cholesterol has a difficult way getting across the cell membrane. It doesn't come across through action of a protein that's specific for cholesterol. Instead, cholesterol crosses the lipid bilayer contained within an LDL. So what happens is LDLs have a protein on their surface that binds to a specific receptor on the cell's surface. And you can see that process happening here. So here's a cell, here's blood supply out here. This LDL has bound to a receptor on the cell surface, and this portion of the cell surface internalizes, as you can see here. So membranes I've talked about yesterday, how they can fuse, how they can bud, and how they can do various things. We see this membrane basically budding off on the inside, and now it contains everything that was in that LDL. Cells have enzymes that will basically take this LDL apart and get the goodies out of the inside of it. The goodies include cholesterol. The goodies include the fats and the fatty acids and things like that. Okay? So this is a very important mechanism for getting cholesterol and other things into cells that have nothing to do with specific protein transporters. All right? This method is called receptor-mediated endocytosis. I'll repeat that. Receptor-mediated endocytosis. And I'll put that in the highlights. I'm not going to spell it all out here, but I'll put it in the highlights for you. Okay? Receptor-mediated endocytosis. So, as a consequence of that, cells now have cholesterol inside of them. It turns out that cells can also make cholesterol. So cholesterol is a very complicated story that we'll talk about later in the course. Okay, so LDL receptors, very important in getting cholesterol into cells. Well, I know at the bottom other important lipids, and I'm not going to talk about transport of these across membranes because that's not my reason for bringing them up. Instead, I want to make you aware of them because, as obviously nutrition majors, the dietetics majors, you need to know something about what the various vitamins are and what they do. So, everybody's heard of the fat-soluble vitamins. There are 4 of them, known as vitamin A, D, E, and K. And they're fat-soluble, meaning that they don't dissolve very well in water. And as a consequence, they get carried in the body through the LDLs. The LDLs are transport mechanisms for fat-soluble vitamins. Okay? Well, we all know that vitamin A is a very important vitamin for purposes of vision. And vitamin A has a very cool property. Okay? So vitamin A has a series of double bonds in it. And vitamin A has a ring structure. In fact, I don't have the... Actually, I might have that up here. Vitamin A has a ring structure, okay, that's attached to a long side chain. Well, this is actually beta-carotene, but this is what vitamin A is right here. If you take beta-carotene, which is a component in carrots, and you cut it in half, you get this guy right here, vitamin A. Okay? So vitamin A has a ring, and it has what I call a side chain that's out there. And if you look carefully at that side chain, you'll see that that side chain has double bonds. And what I've been hammering into your heads with respect to double bonds in fatty acids is that they can be in the cis or trans configuration. They're usually in the cis configuration when they're in a fatty acid. In vitamin A, they're usually in the trans configuration. However, when vitamin A is exposed to light, vitamin A, one of the bonds in vitamin A, will rapidly convert into the cis configuration. It's known as position number 11. Okay? And so we can see this happening right here. Here's vitamin A with all of the bonds in the trans configuration, and here's vitamin A with the bond at position number 11 in the cis configuration. Okay? Well, look at the difference in those two structures. All right? This guy is straight. This guy is bent. Okay? Kind of like you guys are after that whiskey and coffee that you're drinking there, right? You'll get a little bent. So if you're bent, all right, you've got a physically different shape. Well, vitamin A is in our eyes. Okay? Vitamin A is in our eyes, and it's specifically in a protein called rhodopsin that's in our cones and rods. You heard of cones and rods in your eyes, right? Well, something very interesting happens as a result of this change right here. If we look at rhodopsin, rhodopsin is a protein, and in the very middle of this protein is attached this vitamin A molecule. Okay? Because this bond is sensitive to light, it changes the shape of vitamin A. And since this protein is holding the vitamin A, the protein's shape gets changed as well. Well, the change in shape of that protein causes a series of events to happen, which I'm not going to bore you with the details, much as you might like to know them. All right? But this series of events result in the stimulation of a nerve signal. So it's because of this little tiny change in shape that we're able to see the universe. Okay? It's remarkable to think that this one thing right here is what gives us vision. Okay? It tells us that something like as simple as changing the shape of a protein by a very minuscule amount can have a tremendous effect. Okay? As a consequence of this, we can sense through vision the world around us. Pretty cool thing. Okay? There's a protein that bacteria have, some bacteria have, called bacteriorhodopsin. You can stop writing for the moment. I'll just tell you about this protein. Okay? You're not responsible for what I'm going to tell you. But I tell it to you because it's kind of a cool thing. Okay? This bacterial protein called bacterial rhodopsin is very much like the rhodopsin that's in the membranes of our rods and cones in our eyes. And it works in a very similar fashion. That is, when you shine light on this protein in bacteria, the vitamin A changes shape. Okay? It changes shape. So it changes from the linear to the bent form. If we imagine that I am a vitamin A molecule, you can imagine that, I imagine. If you imagine that I'm a vitamin A molecule, and I am I'm in this protein that's in the membrane of a bacterial cell, all right? And the bacterial cell goes out and gets exposed to light. What's going to happen to that vitamin A? Well, it's going to change from the trans, which is the linear form, to the cis. Trans, cis, trans, cis. And the entire time it's in the light, that bond is changing from trans to cis, trans to cis, trans to cis, back and forth. Okay? Now, I'm using my leg in this scheme for a very important reason. The reason I'm showing you that is that what this protein does in the membrane of a bacterium is it kicks protons out of the cell. Remember how yesterday I said that bacteria had a way of kicking protons out? This is one way that a bacterium can kick protons out. This would be a photosynthetic bacterium that is doing this. It's kicking it out. Okay? Well, what it's doing in that process of that is it's increasing the gradient of protons on the outside of the cell compared to the inside of the cell. The cell could use that for energy. Because remember, we needed energy to bring that lactose into the cell. Okay? So in this way, the bacterium is harvesting the energy of light to make something happen. That's pretty cool. All right? Now, later in the term, when I talk about the electron transport system, which I think Dr. McFadden may have talked about a little bit already, I'm going to tell you how you can take that protein and make a photosynthetic fish. Okay? You've got that to look forward to. Okay, questions before I move on? Stunned to silence. Okay, let's move to vitamin D. Vitamin D is a very important molecule. The more that we study vitamin D, the more we begin to appreciate the importance of this molecule for the immune system, for one. It's a very good stimulant of the immune system, and because the immune system is protective against things like tumors and so forth, this is a very important molecule as an anti-cancer compound as well. Vitamin D is made from cholesterol. One of the reasons that we need cholesterol is we need to make vitamin D. Okay? One of the steps of our body in making vitamin D from cholesterol requires ultraviolet light. Requires ultraviolet light. Okay? That's why getting sunshine can be good for you in terms of making vitamin D. It can be bad for you in terms of doing damage to your DNA, so there's a yin and a yang here. But it's good in the overall synthesis of vitamin D. Okay? No, you're not going to know these structures. Don't worry about the structures. Right? Vitamin D is made from cholesterol. Okay? Vitamin D is also important in making strong and healthy bones because vitamin D is involved in controlling calcium and phosphate metabolism. And calcium and phosphate are what you use to make bones. So vitamin D is necessary for healthy bones. People who are deficient in vitamin D have very bad bones. One of the things about, we're learning about vitamin D over time is that we hear about osteoporosis, right? Everybody hears about osteoporosis. Osteoporosis meaning that they've got weak bones, they're falling apart, right? And we look at those old people that have those weak bones and we think, oh, wow, I'm never going to be like that. Well, I hate to tell you this, but the way those people got to be like that wasn't because they were vitamin D deficient when they got old. They were vitamin D deficient at your age. Oh, yeah. So, if you think about this, knowing something about your vitamin D levels is really, really important. If you want to protect against osteoporosis, this is the time to do it right now. Okay, vitamin E. Vitamin E is the vitamin about which we know the least. Vitamin E appears to play some important roles in development, and it may help to protect against chemical damage by something called reactive oxygen species. Okay? Vitamin E is fat-soluble, and we find it in membranes, not uncommonly found in membranes. Its function is not completely understood. Okay? Dr. Moret Traber on the OSU campus is an expert in vitamin E. I should also mention that Dr. Fritz Gombart on the OSU campus is an expert in vitamin D. Okay? And Dr. Donald Jump on the OSU campus is an expert in vitamin A. So those are, if you guys want to go talk to him, you've got that information. All right, the last of the fat-soluble vitamins is vitamin K. And vitamin K plays an important role in the clotting of blood. Dr. Traber also works on vitamin K. Okay? The Linus Pauling Institute, of which all three of those people I just mentioned are members, is very interested in micronutrients and vitamins. Okay? This guy protects, I shouldn't say protects, but it helps the blood clotting process to happen, because it makes some modifications to proteins, and those modifications allow the proteins to bind to calcium. And that binding to the calcium is a critical step in the clotting process. I'm not going to be able to go through the clotting process here, but I'll just tell you that those modifications are necessary. If you block the action of vitamin K, you limit the ability of the body to clot blood. So if you've ever heard of anybody who's on blood thinner, anybody know anybody on blood thinner? A few? Yep. Okay. If you know somebody on blood thinner, what a blood thinner does is it counteracts the action of vitamin K. Okay? The most common blood thinner is a compound known as warfarin. W-A-R-F-A-R-I-N. Warfarin is more commonly known as rat poison because you use it to kill rats. And you think, wow, I never realized I was going to go make a whole bunch of bloody rats in the process of poisoning them. Well, in fact, you don't really do that. You give them a big enough dose that what happens is they hemorrhage internally and die. Okay? So a person who's on blood thinner is on a very weakened dose of that warfarin to reduce the clotting ability of the blood. They don't want to stop the clotting ability of the blood completely, because if you stop that completely, you will hemorrhage and you will die, just like a rat will. Okay? Okay. Questions about those vitamins? Yes? Where can vitamin K be found? It's circulating in your bloodstream right now. Yep. You want it there available to help the clotting process. Okay. Let's move forward and talk about a very interesting class of compounds called the prostaglandins. How many have ever heard of prostaglandins? 1, 2, 3, 4, just a handful. Okay. So prostaglandins are some really interesting and odd compounds. Okay. Prostaglandins are made starting with a fatty acid. So the starting material for making prostaglandins is a fatty acid known as arachidonic acid. Arachidonic acid. And if you look at this, you can see arachidonic acid is a polyunsaturated fatty acid, meaning it has more than one double bond. In fact, it has 4 double bonds. It also has 20 carbons. We call compounds like that eicosanoids. E-I-C-O-S-A-N-O-I-D-S. Eicosanoids are compounds that have 20 carbons and multiple double bonds. Okay? Arachidonic acid is not a prostaglandin. It's the starting material for making a prostaglandin. Okay? You have arachidonic acid sitting attached to glycerophospholipids in your membranes right now. So some of those fatty acids that are attached to the glycerol and the glycerophospholipids, some of those are arachidonic acid. When your body goes to make when it goes to make prostaglandins, it first has to release that arachidonic acid from the glycerophospholipids. So there's an enzyme that will do that. Okay? An enzyme that will do that. The enzyme has a name, and it's the day for names. It's called lipase A. You can call it, abbreviate, you can call it PLA. Okay? PLA will cut arachidonic acid off of a glycerophospholipid in your membrane. Okay? Well, what does arachidonic acid do? Arachidonic acid will get converted into a prostaglandin. These are all different kinds of prostaglandins on the bottom panel. And there's an enzyme called a cyclooxygenase, CYCLOXYGENASE. A cyclooxygenase will convert arachidonic acid into a prostaglandin. Why do we call it a cyclooxygenase? Well, look what we started with up here, and look what's down here. We've made a ring. That's the cyclo part. And the oxygenase part is we've put oxygens on the molecule in various places. It's a cyclooxygenase. All right? Prostaglandins do a lot of different things in the body. Okay? They do a lot of different things in the body. Some of them are involved in pain. Okay? If you go out and get stung by a bee, one of the reasons that you feel pain is that the bee sting is releasing, or I shouldn't say it's releasing, it's activating phospholipase A, which is cleaving off those arachidonic acids, which are getting made into prostaglandins. And the prostaglandins, one of the things that they will do is cause you to feel pain. Okay? How do you counteract that pain? Well, you counteract that pain with a pain reliever. You take aspirin. You take ibuprofen. Aspirin and ibuprofen have a common property. They're known as cyclooxygenase inhibitors, or COX inhibitors. They call these enzymes COX, by the way, COX. You can abbreviate that if you want to. COX inhibitors. If you inhibit the enzyme that makes prostaglandins, and prostaglandins cause pain, then you're inhibiting pain. Now, there's different kinds of pain, but prostaglandin pain is one kind of pain. You take COX inhibitors for a headache because you're stopping or slowing down the synthesis of the, slowing down the synthesis of prostaglandins from arachidonic acid. Okay? Pretty cool. So COX inhibitors are very important that way. Aspirin and ibuprofen are specific inhibitors of the COX enzyme. Okay. Prostaglandins themselves are molecules that are made into other things as well. So we're seeing a sort of chain of events here. Arachidonic acid made into prostaglandins, prostaglandins made into something else. One of the things that prostaglandins are made into, is another group of molecules called thromboxanes. Thromboxanes. Thromboxanes are related to prostaglandins, and they function to make the platelets in your blood sticky. So if aspirin is inhibiting the synthesis of prostaglandins, and prostaglandins are necessary for the making of thromboxanes, and thromboxanes make platelets sticky, aspirin is reducing the clotting ability of blood, just like warfarin, but by a different mechanism. Okay? It's using a different mechanism, but it's also reducing the clotting ability of blood. Now, one last thing I will tell you about the prostaglandins and the aspirin, then I'll move on to another topic, and that is aspirin has another name we give it. It's called an NSAID, N-S-A-I-D. It's an abbreviation for something. You like all these names today? You don't need to know what it stands for, but I'll tell you what it stands for. It stands for non-steroidal anti-inflammatory drug. It means that aspirin is not a steroid. Okay? Aspirin prevents inflammation because prostaglandins cause inflammation. That's one of the ways they cause pain. Okay? Well, if there are non-steroidal anti-inflammatory drugs, there must be steroidal anti-inflammatory drugs, and there are. Okay? How many of you have ever had to have cortisone for something? Okay. Cortisone is a pain fighter and a swelling fighter. And it works on this very same group of molecules as I just described to you, but it works in a different way. I told you that the aspirin was stopping the synthesis of prostaglandin, right? What the steroidal drugs do is they inhibit phospholipase A. They inhibit the release of arachidonic acid from the membranes. So if you have no arachidonic acid, you can't make any of these things. Okay, that's a lot of stuff. That's a lot of stuff. Okay. Maybe this would be a good point to stop and do a song. Yes?
Why do they put some numbing medicine and painkillers in the cortisone shot?
Why do they do what?
They added like numbing medicine and other painkillers in the shot.
I don't have an answer to that. I don't know. I'm not aware of that. No, sorry. Sometimes numbing medicine goes into shots because the actual giving of the shot, like in a Novocaine or something, will actually make the giving of the shot easier. That's be my guess, but I don't know. You guys want to do a song? All right, we'll do a song at this point. I just thought of one that would be useful. All right? You guys know the song Oklahoma? All right, let's go back. I've got 180 songs. You guys are going to hear songs the whole damn term. Ready? All right, Oklahoma. Prostaglandins, the icosanoids creating pain, are the ones to blame when you get inflamed and ouch, they hurt inside your brain. Prostaglandins, every throb and ache gets magnified. If you hope to win, cyclooxygen's generation's got to be denied. The Vioxx has all been recalled, so go get yourself Tylenol. And if you ache, blame PG aches and days. We must complain that you make the aches prostaglandins, prostaglandin, D2F1, G2E2, prostaglandin. It's you. All right. Now you'll always remember prostaglandins by my high voice. Okay. The last thing I'll say is that this class of compounds, I've already talked about thromboxane here. You can see what thromboxane looks like, and again, you're not going to draw it, but thromboxane looks kind of like prostaglandin, kind of an exotic structure with that extra oxygen there. Okay. Thromboxanes, as I said, are involved in making platelets sticky. Another very interesting compounds in this class of compounds are known as leukotrienes, and they're not made from prostaglandins. They're made by a related pathway, but they look kind of like prostaglandins. They're made from arachidonic acid. Leukotrienes are very important in asthma and constriction of bronchial tubes. So one of the strategies for treating asthma is actually to make inhibitors for the enzymes that make the leukotrienes. So important consideration. Okay. That's as much as I want to say about those guys. Let's turn our attention to nucleic acids. Has Dr. McFadden talked about structure of nucleotides and stuff or not? No, okay. So this will be new stuff. All right. When I say nucleic acids, well, I hope the first things that pop into your mind are DNA and RNA. Okay? Because that's what the nucleic acids are. And nucleic acids have, as their building blocks, nucleotides. And when students learn this the first time, they plant it in their head something from high school, and that is, well, nucleotides are A, G, C, T. And in fact, they're not. They're the bases of the nucleotides. So nucleotides have 3 components. The base is one of the components. Okay? So a nucleotide is not an A, a T, or a G, or a C, but the A and T, a G, or C are part of that. So the base, all right, you guys will memorize all those structures? It was a nervous laugh at that point. No, you're not going to memorize those structures, all right? But You should know some general things about them. You should know that one group of the structures are known as pyrimidines. The pyrimidines have a small six-membered ring. The other group of bases is known as the purines. And they have two rings. They're bigger. You should know that the pyrimidines include cytosine, thymine, and uracil. And you should also know that the purines include adenine and guanine. So there are 5 bases that commonly appear in DNA and RNA. And there are some other ones that actually do occasionally occur in RNA we'll talk about later. But that's the 5 bases that appear there. You know, I think, probably, again, from basic biology, that DNA has thymine, and in its place, RNA has uracil. Okay? Both of them have cytosine, both of them have adenine, both of them have guanine. I said that a nucleotide contains 3 things. All right? Let's take a look at a nucleotide. All right? Here are the 3 things that we see in a nucleotide. It has a base. There's the base that you saw right there. It has a sugar that you can see in this 5-membered ring down here. And a nucleotide will always have at least 1 phosphate. The phosphate being over here. Okay? If we look at the sugar is numbered by its carbons. And you can't see it from there very well, but I'll tell you right here. This carbon that's attached to the base is carbon number 1. Carbon number 2 is down here. Carbon number 3 is there. Carbon number 4 is there. And carbon number 5 is above the ring right there, where that pointer is. Okay? I tell you that because we use this numbering to orient ourselves with respect to a DNA or RNA molecule, which I'll explain in just a little bit. When I talk about 5' and 3', that'll relate to that 5 that's right there and the 3 that's right there. Okay? All right. The sugar that's found in RNA is known as ribose. In its place in DNA is a different sugar called deoxyribose. And as its name suggests, deoxyribose is lacking a sugar, lacking an oxygen, not a sugar. It's lacking an oxygen, and specifically it's lacking the oxygen right there at position 2. You see the OH at position 2 of RNA nucleotides, and there's position 2. It only has a hydrogen for DNA nucleotides. Okay? That small change really drastically changes the chemical properties of RNA and DNA. Okay? There's another term I want you to be familiar with, and that's a nucleoside. So I told you what a base was. I told you what a nucleotide was. A nucleoside is something in between. Okay? A nucleoside looks very much like a nucleotide, except for it's missing something. It's missing phosphate. So a nucleoside has a base, and it has a sugar, but it does not have a phosphate. A nucleotide will always have at least 1 phosphate. Biochemists like to refer to nucleotides oftentimes as nucleoside monophosphate, nucleoside diphosphate, nucleoside triphosphate. If I said nucleoside diphosphate to you, I would hope that what would pop into your head is that must be a nucleotide, because nucleoside with a phosphate on it is going to be a nucleotide, right? So a nucleoside monophosphate or a nucleoside diphosphate or a nucleoside triphosphate is the same thing as a nucleotide. ATP is a nucleotide, adenosine triphosphate. So you've heard about ATP. Now you know it's a nucleotide. The energy of cells comes from the hydrolysis, that is the breakdown of ATP. And when they break it down, what they do is they clip one of the phosphates off. That makes ADP. adenosine diphosphate. Okay? Okay. Let's look at how the nucleotides are put together to make a nucleic acid. So a nucleic acid is a polymer of nucleotides. We think about plastics being a polymer of small building blocks. A nucleic acid is a polymer of nucleotides. This polymer is RNA. Okay? Look how the individual nucleotides are joined to each other. Here is one nucleotide right here on the very top. Here's another one that's been attached to it, and we can see that what's happened is the phosphate that was on carbon number 5 got attached to the hydroxyl that was on carbon number 3. Okay? And subsequently down here, the next one had the phosphate on number 5 attached to the carbon on number 3 of the next one, etc., etc. This could go on for thousands or millions of nucleotides. Nucleic acids, by far, are the largest molecules in cells. There's nothing even close to them. People always think about proteins being large. Proteins are large, but they're nothing compared to nucleic acids. Nucleic acids are gigantic. If we take, for example, DNA, this is kind of hard to envision, but it's true. If you take DNA, okay, and you took all the DNA in your cell and you unwound it, because it's all wound up very tightly. If you took all the DNA in one cell, a cell that's smaller than your naked eye can see, and you unwound all the DNA that was in that cell, it would be 7 feet long. Okay? That's remarkable. All right? It says that there's got to be a hell of a lot of winding up to fit that into the confines of a cell, and there is. Okay? All right. This is what an RNA would look like. A DNA would look just like that, except for it would have deoxy, 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 and it would have a complementary strand. You know from basic biology that U pairs with A, G pairs with C, C pairs with G, and A pairs with, in this case, it would pair with T. If that was DNA, this would be a T to an A, etc. You know the rules. Okay. I also show you this figure to show you what I described as the orientation of the nucleic acid. I said that the 5 and the 3 were very important in understanding the orientation. If we look at this, we see that this 5-phosphate got linked to this 3-hydroxyl. Those are no longer free. They're bound to each other. This is bound to each other. This is bound to each other. There's only one part that has a free 5, and that's this guy up here. It's what we call the 5' end. This phosphate is not attached to something else. At the other end of the nucleic acid, we see that this hydroxyl is not attached to something else, and it's what we call the 3' end. When you hear people talking about 5' and 3' ends, that's what they're referring to. Okay? The 5' end will always be not attached to something else, and the 3' end will always not be attached to something else. Well, to add to your nomenclature for the day, I'm going to give you one more nomenclature, and that's right here. This bond that joins these two is a covalent bond. It's a very strong bond. Covalent bonds, you should remember, are strong bonds. Okay? This strong bond that joins nucleotides together is called a phosphodiester bond. PHOSPHO DIESTER. And yes, I will put all these in the notes as well. Okay? Phosphodiester. So a phosphodiester bond links adjacent nucleotides together. Okay. DNA, everybody knows, is a double helix. Everybody knows that G pairs with C, T pairs with A. If we look at the orientation of what I just showed you with respect to that one strand, the phosphates and the sugars are the band part here. where you see the yellow ribbon and you see the purple ribbon, that's where the phosphate and the sugar is located. It's on the outside of the DNA molecule. On the inside of the DNA molecule, that's where the bases are located. Okay? This structure was determined in 1953 by Watson and Crick, I started to say Clark, Watson and Crick, using data they stole from a woman named Rosalind Franklin, a very sad but true story. She was the genius that actually was probably the most impressive crystallographer of her era. Her data was taken from her by Watson and Crick. She did not get the Nobel Prize, partly because she died. There's a long story associated with that, but it's a sad story that was her data that they used to make this determination. Okay. Base pairing, up close and personal, looks like this. This is how it is that adenine pairs with thymine. Hydrogen bonds. Dr. McFadden has talked about hydrogen bonds, I trust. Okay. Hydrogen bonds are weaker than covalent bonds. They are attractions of partial charge to each other. That's what hydrogen bonds are. We look at the bonds between adenine and thymine, and we see there are only 2 hydrogen bonds, whereas there are 3 hydrogen bonds between guanine and cytosine. And if I had uracil and adenine up here, I would still only have 2 hydrogen bonds. Okay? That means that the forces holding together AT base pairs are not nearly as strong as the forces holding together GC base pairs. That will become important when we talk later about transcription and DNA replication. Okay. There are 3 common forms of DNA that we find in cells. The most common form that we find is the one that was described by Watson and Crick, and that was the B form of DNA. It's the one that's in the middle. Okay? And the B form of DNA has about 10 base pairs per turn of the helix. About 10 per turn of the helix. It looks like a spiral staircase. And in fact, you can see that the steps are pretty flat. You can imagine that if this were blown up to the size of this room, you actually could climb that like it were a spiral staircase. And you could. The other 2 forms of DNA are not so friendly for climbing, as it were. The form on the left is called the A form of DNA. And it was actually the form of DNA that was discovered by Rosalind Franklin, and she got credit for that. Unfortunately, it was a relatively minor form of DNA that at the time she discovered it was thought not to be important in the cell. We know today it actually has some importance, and we know that it actually exists. At the time they thought it was a fluke and it wasn't something that was real. But Rosalind Franklin discovered this form. You look at this, you wouldn't want to try to climb that spiral staircase. It's twisted. Okay? It's also scrunched down compared to the B form. The third form was discovered in the late 1970s. It's a form called the Z form. It was called Z because, at least in some ways of looking at it, it makes a zigzag. That's what it looks like to people. Okay? The Z form of DNA is fundamentally different than the A and the B forms. Even though the A form looks different than the B form, they have a lot of similarities. Z is very different. You can see it's stretched out. And there's something else about the Z form that's kind of different. In fact, when people first saw Z, they said, no way that's real. No way that thing forms in the cell. Well, one of the things you learn in biology is never say no way, because they later discovered that it did. Right? What's so odd about it, besides being stretched out, is the fact that instead of the 2 strands twisting in one certain way, they twist in the opposite way. Okay? Strands turn out to have a handedness associated with them. We call one form right-handed and the other form left-handed. Both the A and the B forms are right-handed. The Z form is left-handed. If you're curious to see what a Z and a B and A form, what left-handed, right-handed means, come by my office sometime and I'll show you. Okay, I won't go through it here. And no, you're not going to have to look at something and say that's left or right. But you should know that A and B are right-handed and that Z is in fact left-handed. Okay. By the way, the B form probably exists about 99% of the time. It's by far the predominant form that's found in cells. Okay. That's trying to show you, it doesn't do a very good job of telling you what right versus left is. Supercoiling. Okay? Supercoiling. When you take DNA strands and you start twisting them around each other, what you discover is that you can twist them too much or you can twist them too little. Remember I said we had about 10 base pairs per turn. If I twist them so that they've got about 11 base pairs per turn, it's like taking a rubber band and twisting it. You put that tension in a rubber band, what's going to happen to it? Well, it's going to relieve that tension in some way if it can. Okay? The same thing happens with a DNA molecule. If I cause some additional twist to be put into this thing, and then I close it up so it can't relieve that tension, like that rubber band couldn't relieve that tension, what's going to happen to the rubber band? Well, it's going to all shrivel up. Try this. Take one side of a rubber band, hold on to it, and take the other side and twist it a few times, and you'll see it'll just shrivel all up. That phenomenon is known as supercoiling, and it's depicted schematically right there. You see that instead of lying there nice and flat and behaving itself, it's all scrunched up. And scrunching it up actually helps the DNA to fit into a smaller space. It's one of the ways we can decrease the size of a DNA. Okay? So supercoiling happens when we change the number of base pairs per turn away from 10. It happens if we put too many twists in or too few. Exactly the same thing happens, because in both cases, it shrivels up. Okay, let's see. Oh, I've got a couple minutes yet. I told you that if you took all the DNA from your cell and you laid it end to end, it would stretch 7 feet. Okay? It says it's got to be coiled up. In fact, if you took all the DNA out of every cell in your body and you stretched it end to end, okay? So you took every piece of DNA in your body and you stretched it end to end from every cell, it would go to the sun and back. Folks, you have 180 million miles of DNA walking around with you. Who says you're not important? Now you know why you don't like to get up on Monday morning. You got 180 million miles of DNA to wake up, right? Maybe that you need that bourbon there, right? Well, the way that we fit DNA into cells is that we use proteins to coil it up. I can take 500 feet of kite string and hold it on a very tiny spool, right? So similarly, if I spool the DNA, I can make 7 feet of DNA go into a much smaller space. I have to do that in order to make it fit. Okay? Those proteins that I use to coil up the DNA are called histones. Histones. The DNA wraps around it. The histones in this figure are the sort of purplish structures. The DNA is the green. And you can see that this DNA is really coiling up. In fact, to make these coils, the coils themselves will start coiling. It gets very complicated. When you see a chromosome in a light microscope, you're not seeing DNA. You're seeing a complex of DNA and protein. That complex of DNA and protein is called chromatin. Okay? So if I take histones and I wrap DNA around it, I make chromatin.